In this tutorial, we'll navigate through the Loupe Browser version 7, which has a brand new user interface and a new multi-sample differential expression feature. You will learn how to visualize single-cell RNA sequencing data, explore gene expression patterns, identify cell types, and conduct differential gene expression analysis across experimental conditions. We will also introduce the Reanalyze function. If you have not updated to version 7, check out the link to our tutorial for the older version below in the description. We will be using the lung cancer tutorial dataset, which you can download from the link in the Loupe Browser. This dataset contains the results from 21 flash-frozen tissues from treated and untreated non-small cell lung cancer and normal adjacent samples from a 10x genomics dataset. Each dot we see here is a cell. The cells are clustered based on their gene expression similarity. To pan or reposition the plot, simply drag your mouse on the plot. You can also use the mouse wheel or trackpad to zoom in and out, or the zoom bar down here. But for now, we will stick to auto scale. Finally, cluster labels appear as you move your mouse over the plot. In the Loupe Browser, the default projection is a t-SNE plot. t-SNE plots take a high-dimensional dataset and converts it to a low-dimensional clustered graph while retaining much of the original information. The pipeline will generate a UMAP plot as well. UMAP is a newer technique, and just like t-SNE, it is a method to visualize high-dimensional data in a 2D space. However, UMAP runs faster, and it also better preserves the data's global structure. If you look at these two clusters, for example, you can see that on the plot they are close together. In UMAP, this actually means that they are more similar. But in t-SNE, the distance between clusters has no meaning. In order to export the plot, just click the Export button on the top right corner. It gives you the option to either export as PNG or SVG. This is an extremely useful tool for saving the plots. Right now, we are looking at the graph-based clusters, which are automatically populated when you open a cloop file. You also have the option to use k-means clustering and define the number of clusters. This dataset also comes with a disease state category and a sample ID category. We can go up here to split view by disease state. Moving on, we will now click Run Differential Expression. If you have version 7.0 or later, you'll have 3 different options for running differential expression. The first compares selected clusters to the entire dataset. The second compares and distinguishes each checked cluster with the other currently checked clusters in the group. The third compares selected clusters across multiple samples between 2 conditions using pseudobulk. We will be going into detail with each of these options. First, we will find the genes that distinguish the untreated cancer from the rest of the dataset. Select disease state, then click Run Differential Expression. Next, click the drop-down and make sure you have 2 entire datasets selected. Now start the analysis. This may take a while depending on your computer. The results of the analysis now appear in the data panel. The table shows significant features, their log2 fold change values, and their p-values. You can see that KRT14, KRT5, and S100A2, all basal cell markers, are significantly upregulated in the untreated cancer group compared to the other 2 groups. Clicking on the gear allows you to change the table settings. We are currently filtering features by upregulated genes, but you can also select downregulated or all significant genes. Also, we usually unhide the genes with low average count. You can click here to export a selected number of features to a CSV file. The analysis also generates a heat map. Each column represents the level of expression of a significant feature, while each row represents a cluster. Grid cells are colored by a gene's log2 fold change compared to the other clusters. Hovering over the columns will show the names of the features represented. Here you are able to export the heat map as either a PNG or SVG. In this comparison, we saw that many basal cell markers were upregulated in the untreated cancer group. This example also comes with a pre-annotated group called azimuth annotations. Unselect all other groups and select basal. Now the plot shows the distribution of basal cells split by the 3 different disease states: untreated cancer, treated cancer, and normal adjacent tissue. Basal cells are thought to give rise to squamous cell lung carcinoma, and it is clear that there are fewer barcodes annotated as basal cells in the normal adjacent tissue group compared to the 2 cancer groups. We will now dig deeper into the features that distinguish the basal cells of treated versus untreated patients, excluding the normal tissue group. To do this comparison, we need to create custom groups. Go to the Advanced Selection tab on the left and click Create New Rule. Choose in basal and in untreated cancer, then save the barcodes to a newly created comparison group, which we'll name Basal Comparison. Then repeat to create a basal treated group. This time we will run differential expression between selected clusters. Here in the table, you can see that PTHLH, which is secreted by certain kinds of squamous cell lung cancer cells, is expectedly downregulated in the treatment condition. To look more into the PTHLH gene, you can add it to the feature list by clicking on the 3 dots. On the UMAP, you can see the PTHLH gene distribution. There are 3 different scale values available: linear, log2, and log norm. When linear is chosen, actual UMI counts are rendered in the view panel without any normalization. Log2 is the default setting and displays log2 transform rendering of UMI counts in the view panel. Selecting log norm as the scale enables viewing of feature expression normalized by UMI count for single-strand spatial datasets or cut site count for attacked datasets. Once log norm is selected, you can view the Violin plot of the PTHLH gene by comparing basal untreated and treated. This plot is also exportable. Finally, we'll explore multi-sample comparison. Version 7.0 and later allows users to find features that are differentially expressed using a pseudobulk calculation. A pseudobulk is formed by aggregating the expression values from a group of cells from the same sample. We will be comparing basal cells of treated versus untreated patient samples, this time using multi-sample analysis. Please note that this method is only suitable if you have biological replicates for each condition, similar to bulk RNA-seq. If you only have one sample for each condition, we recommend using the comparing between selected clusters method instead, which was described earlier. Now click the basal group of azimuth annotations, then run differential expression. Select the Across Multiple Samples option from the dropdown and start the analysis. A separate window will pop up. First, choose Azimuth Annotations in the Cell Types selection dropdown. The clusters are auto-populated, so delete the rest of the clusters and keep the basal cell cluster. Next, select the disease state in the Experimental Conditions dropdown. Select treated cancer as condition A and untreated as condition B. Select Sample ID in the Sample ID section, and the conditions will be auto-populated. As of now, we do not have any issues using basal. However, please note that if a particular sample does not have any barcodes assigned to one of the cell types selected in Step 1, the sample will be highlighted and the multi-sample comparison will not proceed. For example, if you add CD8 T cells to the top, you can see that many samples become highlighted in yellow, which will stop you from being able to move forward. In order to run the analysis, you must delete the highlighted samples, but for now we will only keep the basal group. Now you can see the resulting feature table. You can see that the PTHLH gene is downregulated in condition A, the treated cancer group. If you have your own marker genes, you can import them to do custom cell typing. Here is an example feature list. The first column is the group or type of feature, whereas the second column is the name of the feature. Excel files with the proper formatting can be imported to create a new feature list. You can see that 5 groups of cell type markers have been created. It is also possible to create a custom feature list via the search option in feature mode. We will add a few more features to the basal group, KRT15 and SFN. Make sure the basal group is selected, then search the first gene feature. Clicking on the gene name adds the gene to the selected feature group. Repeat this for SFN. Assuming that we are interested in just the basal cells within the basal comparison category, select Reanalyze. You can see that the 6,941 cells are kept. We can also threshold by UMI count, features, and mitochondria content. We'll set a minimum feature count of 100 and a maximum feature count of 10,000 features per barcode. The next step is to filter cells by mitochondrial fraction, which is the percentage of UMIs per barcode associated with mitochondrial genes. Overexpression of mitochondria genes could signify dying cells. We can select the human reference genome, submit it, and set the mitochondrial fraction maximum of 5%. This threshold will vary depending on your experiment. Select to generate both t-SNE and UMAP plots, name the reanalysis basal only, and click Recluster. In the background, the Loupe Browser will run the same principal components, clustering and t-SNE and UMAP algorithms as the Cell Ranger pipeline. It will only take a couple of minutes. Once the reclustering is done, we can review the t-SNE and UMAP results in basal only and do a similar analysis that we described in this video tutorial. If you have any questions or are interested in single-cell sequencing services, feel free to email our team at support@singulomics.com and check out our website linked in the description.